Hi everyone, I'm Xuexiao from the Center for Reproductive Medicine, Shandong University. In this chapter, I'm going to talk about the immunophenotyping and cell sorting. Immunophenotyping. The phenotypic and functional analysis of immune cells includes the detection of cell type, phenotypic characteristics, cell activation, cytokine production, transcription factors, and other functional molecules. The multicolor fluorescent staining to label more markers on a single cell allows phenotypic and functional assessment of multiple subsets in complex cell populations at the same time. It helps to discover and analyze real cell subsets with small amount of samples. So it's suitable for analysis of samples from reproductive tissues or cells. Here we take the immunophenotyping in the maternal-fetal interface as an example. The maternal-fetal interface is the basis of immune tolerance in early pregnancy. The maternal-derived decidual cells and immune cells and the fetal-derived trophoblast cells, these cells interact with each other to maintain normal pregnancy. An in-depth understanding of the immune response in the periphery and at the maternal-fetal interface during pregnancy can reveal the mechanism of maternal-fetal immune tolerance and contribute to elucidate the pathogenesis of pregnancy disorders such as miscarriages, preeclampsia, and preterm birth. In this study, by comparing the NK cells from peripheral blood and decidua in early pregnancy, they found that the decidual NK cells highly express CD49A and EMOS. The decidual NK cells can also secrete growth-promoting factors to promote embryo development, such as pleiotrophin. The immunofluorescence data were consistent with the flow data. In addition, they found that in patients with recurrent pregnancy loss, the number of CD49A positive decidual NK cells was reduced, and their ability to produce growth factors was also decreased. Therefore, by using multi-parameter flow analysis, they found new immune subsets with specific characteristics and function, which involved in the pathogenesis of pregnancy loss. Some flow cytometer can sort cells into predetermined subpopulations. Cell sorting, also known as fluorescence activated cell sorting. Sorting is the process of physically separating a cell from a population based on some measurable characteristics. To perform flow sorting, you need to prepare the single-cell suspension and then stain the cells with fluorescent antibody to make it ready for flow. If the cell meets the sorting criteria, an electrical charge is applied to the liquid stream, which is being accurately broken into droplets containing the cells. This charge is applied to the stream at the precise moment the cell of interest is about to break off from the stream, then removed when the charged droplet has broken from the stream. As the droplets fall, they pass between the two metal plates, which are positively or negatively charged. Charged droplets get drawn toward to the metal plate of the opposite polarity, and then deposited in the collection tube or onto a microscope slide for further examination. Flow sorting has high accuracy and high speed, up to tens of thousands of cells per second. Multiple cell subsets can be sorted at the same time. More importantly, the sorted cells are viable and can be used for culture, mouse injection, DNA-RNA protein detection, and also for cell therapy. Recently, with the development of high-throughput omics technology, flow sorting combined with transcriptome and single-cell sequencing could explore cell characteristics on both single-cell level and molecular level to elucidate the heterogeneity of disease pathogenesis. Here are some examples. In these two studies, ovarian tissue from female adults or fetal gonadal tissue were collected and prepared for cell suspensions. Then specific cell populations were fact sorted for single-cell transcriptome sequencing. In the first study, the 7AD positive dead cells were depleted by flow sorting to ensure the cell viability for sequencing. While in the second study, the CD117 positive germ cells and CD117 negative somatic cells were flow sorted separately to enrich these two populations for sequencing. In this study published in 2018, the placenta, decidua, and peripheral blood during early pregnancy were collected and prepared for single-cell suspensions. Then the cell sorting was performed. The CD45 positive immune cells and the CD45 negative non-immune cells were individually sorted and then mixed for 10x genomic sequencing. This strategy is to artificially enrich the proportion of immune cells for sequencing. At the same time, 12 different immune subsets were sorted for SMART-6 single-cell sequencing. The accurate sorting of different immune subsets is to artificially enrich the rare cell population with low frequency for effective sequencing and analysis. In this study, 3 decidual NK subsets with different immunomodulatory effects were identified by the single-cell analysis. The characterized molecules screened by the single-cell data were further validated by in-depth flow analysis. And then the morphological analysis of the 3 decidual NK cells was performed after cell sorting to further validate the accuracy of cell typing. This study confirmed the relationship between maternal immunity and placental implantation, providing a basis for treatment of pregnancy disorders such as preeclampsia and miscarriage. Therefore, flow analysis and cell sorting is very helpful for isolation and enrichment of cell population with specific phenotypes. Okay, in this section, we introduced the application of flow cytometry analysis and cell sorting in clinics and also in basic research on reproductive medicine. Hopefully, you can apply it in your own research on reproductive medicine. Thanks very much.